exam will be. It's not worth 10 points. It's not the whole exam, but it's going to be part of it. Okay. So we left off on Tuesday talking about the cell wall. And we talked about all of those differences between gram-positive and gram-negative. We talked about atypical cell walls. What is the difference between mycobacterium and mycoplasma?
Mycobacteria use the acid-fast stain.
Yes.
Mycoplasmas don't have a cell wall.
Bingo. Mycobacterium, acid-fast stain because they have mycolic acids. Mycoplasma, no cell wall. And remember, that was another one I told you a lot of people get those mixed up. So I really tried to emphasize that. So it's, again, I know that it sucks to sit in a class for an hour and 15 minutes, but it's an hour and 15 minutes. You've just got to pay attention because I will say things that are not on the slide and I do expect you to know them. Most of the time they are on the slide, but I still expect you to know them. Okay? So you've just got to pay attention. I know. But you got to do it. Okay. Damage to the cell wall. Really classically, we're looking at things like penicillin and other antibiotics that inhibit or interfere with cell wall biosynthesis. And that is such an important target for clinical applications because the cell wall doesn't exist in us. We don't try to make anything like a cell wall. We don't have proteins or enzymes at all homologous to the proteins and enzymes used to make a cell wall. So if we interfere with the cell wall, that's a magic bullet target because it should not interfere with anything in the host, only the microbes. So penicillin inhibits the ability for microorganisms to successfully build their cell walls. If you can't build a cell wall, right, they eventually will lyse because of osmotic pressure. Remember, really importantly, for those bacteria that have a cell wall, one major function of the cell wall is to help them maintain their structure and maintain their integrity under conditions of osmotic pressure. It keeps them from bursting. When you don't have a cell wall, you are probably going to burst. But a lot of times when we look at gram-positive versus gram-negative, penicillin and its derivatives are not our first choice antibiotic for gram-negatives. That's because they have that second membrane. The second membrane helps them last longer in the absence of a cell wall. They can sometimes last hours without a cell wall. And that's basically equivalent to you missing one dose of antibiotics. They just have to last until the concentration of antibiotics in your body gets low enough, they'll start replicating again. They just have to outlast you. And they will. They're very good at it. So it's really important, right, to think about those of you going into clinical medicine, do the penicillin derivatives you have or that you're thinking of prescribing work against gram-negatives? When we talk more about various antibiotics and things like semi-synthetic antibiotics, we'll talk more about how scientists actually modify penicillin to make it more effective for gram-negatives. But penicillin itself, not great against gram-negatives. So I think I've already answered this, but again, it's something for you to think about because I love asking questions about you applying the knowledge you've learned, not just, can you tell me what penicillin does to the cell, but why would we use something like penicillin? Why is the cell wall such a good target? Right? It doesn't harm human cells. We don't have peptidoglycan. Who cares? Doesn't hurt me any to not be able to make peptidoglycan. I've spent 42 years not making peptidoglycan. 2 weeks isn't going to hurt me. Okay? And however old you are, you spent that many years not making your own peptidoglycan, so you'll be fine. Other compounds that can damage the cell wall include things like lysozyme. Now, lysozyme is really cool. This is one of your own body defenses. You make lysozyme. It's a really common component in things like saliva, tears, other bodily secretions, because it can help get rid of those microorganisms. It can also be challenging, though, for some of you going into clinical medicine. One of the things you have to consider when you're taking, let's say you think a patient has strep throat, you're going to take a swab of the back of the throat. If you get saliva on the swab, the saliva has lysozyme that can destroy the streptococcus, so it would turn out a negative test. So you always have to be really cognizant of what type of microbe could be present and what type of combat we might have that could interfere with your results. And so the way that lysozyme works is that lysozyme works by breaking down the bond between the two carbohydrates, the N-acetylglucosamine and the N-acetylmuramic acid. It breaks that bond right here. But again, thinking about the cell wall and how it's put together, we talked about the archaeal cell wall as well, that it's made of N-acetylglucosamine, but it has a different carbohydrate here. So it is a disaccharide, but it's a different disaccharide. And the bond is different. And this bond is resistant to lysozyme. So it's very good that we don't have any archaea yet that we know of that cause infection, because our base line of defense against the cell wall would be non-functional against an archaeal infection. One of the other things to think about is the difference between how something like penicillin and something like lysozyme work. So I told you that penicillin interferes with the cross-linking, the building of the cell wall. What that means is that a cell has to be actively replicating in order for penicillin to work. If you are not trying to build a cell wall, who cares if you can't build a cell wall? I don't want to build a house. You can take the hammer. Doesn't hurt me any. So the other issue is that bacteria can stop replicating. They can go dormant. And again, they can outlast you. So most antibiotics actually only work when the cell is actively replicating. So that's a challenge. However, something like lysozyme, it breaks the bond. The bond already exists. So lysozyme can work against even a cell that's not replicating. And that has, some clinical importance. If a cell has to be replicating, sometimes cells replicate very slowly. A lot of bacteria are very slow organisms. They're so slow that you have to have a patient take antibiotics for 6 to 9 months. So it's really important that you're able to start to make those connections. What type of antibiotic is this? What type of infection is it? Knowing the basic characteristics, because that will really help you treat your patient as effectively as possible. If you have a patient that has tuberculosis and you're like, I'm going to give you a 14-day course of penicillin, your patient's going to die. Well, because one, they have the mycolic acids. Penicillin, not a good choice. Two, they take about 24 hours to replicate. You need 6 to 9 months bare minimum of antibiotic therapy. Right? So that it becomes very, very important to understand these differences and to think a lot about them in those contexts, for those of you interested in clinical medicine. So that's the cell wall. Now we can start to move inside the cell. So remember, the cell wall is one of those bonus features. A lot of cells have it. I would argue most cells, most bacterial cells have a cell wall, but not all, right? We use that classic example of mycoplasma. The plasma membrane everybody's got. You have to have a plasma membrane. We have not yet discovered a bacterial or archaeal cell that doesn't have a membrane. We probably never will. The membrane is kind of what makes the cell. So the plasma membrane. And again, depending on what type of cell we're looking at, maybe we have one, maybe we have two. So what they're showing you here is a cross-section of a gram-negative cell because they're trying to indicate that there are two membranes. They're showing the plasma membrane. Sometimes people call it the inner membrane. Doesn't matter to me. That thin layer of peptidoglycan, and then here's that outer membrane. So that's how I know it's gram-negative, because it has two membranes. Yes?
So if it always has two membranes, it won't always be gram-negative?
Yes. Yeah. And the outer membrane, I know it's a little bit hard to see because TEMs are not perfect, but the outer membrane is a little bit fuzzier and the inner membrane or the plasma membrane is a little bit tighter. Remember, that outer membrane of those cells is not just the phospholipid bilayer, it has all that LPS that hangs out. So it's not as tightly packed, it's not as compact, it can extend pretty far because of all of that LPS. So that's why it looks a little bit more like fuzzy and wavy. But their phospholipid bilayer looks a lot like our phospholipid bilayer. It's a phospholipid bilayer. It's good organization. It has been maintained throughout evolutionary history. Okay, so you have your polar heads that face in and out, like that, inside the cell, outside the cell. You have the non-polar fatty acid tails that are in the middle, hydrophilic, hydrophobic, polar, non-polar, like whatever you want to call them. But of course, the membrane is more than just the phospholipids. It consists of proteins. So there are peripheral proteins. Peripheral proteins are associated with the membrane, but not going through the membrane. Transmembrane proteins will go through the membrane. And the membrane itself is also not what we would call a static structure. So if a protein is right here in the membrane, it doesn't stay here. It can move throughout the membrane. Sometimes it can flip and go, in and out. Protein subunits can come together and form receptors. So it is not a static system, it's a dynamic or a fluid system. Things move. And again, when you lose that ability to move, that becomes a problem. And we talked about that in the context of bacteria and archaea that live in colder environments. You start to lose that fluidity, so they increase the amount of unsaturated fatty acids to retain fluidity, so the fatty acids don't pack so tightly together. The phospholipids themselves are capable of moving, and it can be in some ways self-sealing. So it can seal like tiny injuries or tiny pores that might form. But once you damage it enough, like you can reach that tipping point where it can be completely damaged. So it's a moving, living system on the surface of these cells. Its main function, right? Keep everything in. Keep the things in. If you don't have a membrane, you don't have a cell. So keep the things in, but also, keep the bad things out. We let in what we want to let in. So the membrane is a selective barrier. Some things can pass through it and some things just can't. Depends on the size of the molecule, whether or not it's charged, whether or not it's polar. All of those things can affect movement through the membrane. Now, this is not to say that you can't bring something big and charged and bulky through the membrane. You absolutely can. but it doesn't pass through the membrane directly. It usually needs a protein pore to come through the membrane. It needs an opening that forms to allow it in. So it's very selective about what it lets in. It's like wherever you live, right? You have windows, you have doors, you can open them if you want. You can let things in if you want. You can let things out if you want. But for the most part, there's very little that just freely flows in and out of your home. Very little flows freely in and out of your cell. In bacteria and archaea, the plasma membrane is also where they will have all of the proteins necessary for the electron transport chain, if they have the electron transport chain. Where do eukaryotes store those proteins? Eukaryotes have the proteins, but where do they put them? The powerhouse of the cell, the mitochondria, right? So that's another one that sometimes people will say, oh, you know, the prokaryotes do the electron transport chain in their mitochondria. They don't have one. Remember, they don't have them. No membrane-bound organelles. Prokaryotes do the electron transport chain in the plasma membrane. Eukaryotes do it in the mitochondria. So the plasma membrane does a little bit of extra heavy lifting in prokaryotes because it has to be the site of ATP synthesis for the electron transport chain. We also have a good deal of bacteria specifically that are photosynthetic. So they keep the pigments necessary for photosynthesis also in their plasma membrane. Where do eukaryotes keep those? Not the mitochondria, but very similar in structure anyway. Chloroplasts. And again, I'm highlighting this because one, people will miss it and I don't want you to. I want you to get it right. But two, because this also starts to explain how the mitochondria and chloroplasts evolved from bacteria. That's exactly right. It's the Endosymbiotic Theory. The idea, and we'll talk more about it, but the idea that mitochondria and chloroplasts were once free-living bacteria that were capable of doing respiration or photosynthesis. So they do it in their membranes. Eukaryotes either use the mitochondria, so almost all eukaryotes have a mitochondria, at least one. And some eukaryotes, those that do photosynthesis, have chloroplasts. So that's where they do that. But prokaryotes do not have membrane-bound organelles, so their plasma membrane does everything. Now, it folds in a little bit, which looks a lot like the thylakoids that we see in the chloroplast. So they use these kind of infoldings as their site for photosynthesis, but it is the plasma membrane. Okay. So the plasma membrane is really, really critical. And then I guess lucky for us, unlucky for them, the plasma membrane can also be damaged. And so when we can damage the plasma membrane, right, that can allow us to target infection. But we have to be very careful because we also have a plasma membrane. So many times these compounds that target microbes in higher concentrations can also target our cells. They might preferentially target microbes, and because microbes are typically smaller than our cells, they have a different surface to volume ratio, they might experience the consequences more quickly. But we do have to worry. So alcohols, the reason that you can use alcohol as a disinfectant is because it can target their membrane. They'll dry out really quickly. detergents disrupt the membrane. Polymyxin is a class of antibiotics that also preferentially targets the bacterial membrane. But polymyxin, remember, we have plasma membranes too, so we have to be careful. So how do we use an antibiotic that could also target our cells? We only use it topically. You would never be given polymyxin as an oral or an IV antibiotic. It's a component of Neosporin. You don't eat Neosporin. Don't eat Neosporin. You put it on a wound. Okay. It's too toxic because it could start to target our cells too. So we can damage the membrane, and that causes the cell to basically fall apart. All the contents start leaking out. It loses the proton motive force. The cell will die. So it's a good way to get rid of cells. Questions to this point? Okay, so plasma membrane, super important. We do have to move things across the membrane. So how do we do that? There are a couple of passive processes. They don't involve any energy use. And we do the same thing. So this is probably something that you've covered in other classes. So simple diffusion is the easiest. You have very small molecules that are at a higher concentration on one side of the cell versus the other. And they will travel from that area of high concentration to an area of low concentration until there is equilibrium. They're usually like small, non-polar, can't be charged, not very bulky, and they can kind of just go in and out as the concentration requires for equilibrium. So simple diffusion. The next is facilitated diffusion. This is where maybe the molecule is charged, maybe it's a little bit bigger, but it's still at a higher concentration on one side than the other. And so we want to reach that equilibrium. And so we can use proteins embedded in the surface of the membrane or embedded through the membrane to allow that to happen. We have some non-specific transporters, things like a chloride ion transporter or a sodium ion transporter, or it might be like cations, so it could do like sodium and potassium and calcium. Right? They're charged, so they won't go through on their own, but it doesn't require any energy because they'll go from high to low. Sometimes they're a little bit bigger, so things like glucose might require a larger pore, but at the end of the day, almost always there's more glucose outside than inside, and you want as much glucose on the inside as possible, so you'll continue to bring it in. Okay. But it doesn't require any energy because these molecules are going from an area of high to low concentration. But on the other hand, sometimes we may want to move other stuff. So we can move water. Maybe we don't necessarily want to move the solute in and out. Maybe we can move water in and out to reach that similar type of equilibrium. It's osmosis. An area of like a higher concentration of water molecules or like a higher concentration of solute molecules. You can bring water to dilute that down, or you can expel water if you have like a really low concentration to make that concentration higher. And so bacteria also have things called aquaporins, proteins that allow water to go in and out. And their osmotic potential is very, very important to them. And we as humans take advantage of their osmotic sensitivity, especially with things like food preservation. So when you have a microbe or any cell where you have equal solute concentrations inside and outside, the water won't move. Not that the water won't move, but that it will always move, but it kind of just moves at an equilibrium. We call that isotonic. But sometimes we have a situation where the concentration of solutes outside the cell is lower than inside the cell. So what we would want to have happen is we'd want the solutes to come out and balance, but what instead happens is the water rushes in. And when the water rushes in, right, that causes the cell to swell. The cell wall is really good at helping that stay intact, but eventually everybody reaches their breaking point and the cell will burst. So a cell placed in a hypotonic solution, a low solute concentration solution, the water will usually rush in and cause the cell to burst. Now, the principle that we take advantage of often with food preservation is hypertonic solution. We put a boatload of solutes on the outside of the cell, which causes water to rush out, and it causes this phenomenon called plasmolysis or plasmolysis. The cell shrinks and is completely dehydrated and then no longer viable. Can you think of an example where you add a lot of solutes to a food to preserve it?
Is it salt?
Salts. Salts are really common, absolutely. Yeah, that's why like beef jerky is salty. This is like the principle why salt was so important. We didn't have refrigeration, but if we had salt, we could prevent the microbes from growing and causing our food to rot. Food doesn't rot if there are no microbes. I prefer sugar, on the other hand. I like sweets. Dried fruits. You have that, you don't have to raise your hand, but I know some of you got that bag of dried mangoes from Costco in your pantry. You had that for a while. You know what? You crack it open, it's still good. Because the sugar dehydrates not only the fruit, sure, but it's really the water in the fruit and the water in the cells that allows them to grow. So things like salts and sugars are really common for food preservation.
I just have like a question because I'm curious. Does fermentation for like pickles or kimchi rely on the same principle as this?
So fermentation is a little bit different. That's because some of the byproducts of fermentation actually can inhibit the growth of the other microorganisms. So that can help with the preservation. But usually like with kimchi and pickles, you also want to put them in an airtight container. But yeah, you can get contaminating organisms in there. And so sometimes having it be either really salty or kind of like the acetic acid can inhibit the growth of some of those spoiling organisms to allow you to preserve it. Yeah, definitely. So this is really important. And you know, the other thing to think about is there are microorganisms that live in deserts where there is very little water. So they have a lot of evolutionary adaptations to allow them to avoid being all dried out. Okay, so those are all processes that don't require energy. What does require energy is active transport, moving a molecule against the concentration gradient. There's very little outside, we have a good amount inside, but we want more. I want more. So that requires the expenditure of energy. You can't bring something in without giving something. So this will also require a transport protein and the expenditure of energy. So again, the classic is just this active transport where you can have a transport protein, you expend some ATP, and that can grab the substrate from the outside and bring it in against the gradient. You can also have group translocation, so this is a little bit trickier. So maybe you have enough glucose on the outside of the cell, or I'm sorry, you have enough glucose on the inside according to equilibrium. But you want to bring more glucose because you need glucose. What you don't have is inside the cell, you don't have very much of this glucose 6-phosphate because to the cell, those are very different things. So you do expend energy because you have to get that phosphate from somewhere, but you add the phosphate to glucose And then the cell is like, oh no, this is different. I will bring this in. I don't have enough of this. So you kind of trick the cell. So it's group translocation. And sometimes they'll even couple like a group translocation where they'll let something out and something else can come in. Just because like, basically again, like the cell kind of got tricked. It opened up the pore to let something out. These are anti-porters. It let something out and then something else just snuck in. Or vice versa. These are like I let the dogs out and then I let them back in and the cat tries to get out. I'm like, no. But it's like that. The cat sneaks out because I'm paying attention to the dog. Because the dog is the thing I want. So I get rid of or I bring in the thing I want and then the other thing can sneak and go in the other direction. But again, a lot of this requires the expenditure of energy. So questions about the membrane before we move more into the cell. More in is where we get the cytoplasm. This is not a bonus feature. Everybody's got the cytoplasm. It's the stuff within the plasma membrane. It's about 80% water, which again is why it's so critical that a cell not be dehydrated. And this applies to our cells too. This is not microbe specific. There are proteins, carbohydrates, lipids, ions. In microorganisms, right, you'll have the chromosome in there. You may have plasmids. The ribosomes are floating around in there. You might have a couple other things like inclusions. And depending on the cell, right, you might have some cytoskeletal components. You may have a cytoskeleton, like the internal skeleton of a cell. With bacteria, and so this part, I'm not going to expect you to remember like all of the names and what they do. I'm just showing you here how some bacterial structures are analogous or homologous with eukaryotic structures. So some proteins some bacteria have protein homologs to eukaryotic cytoskeletal components. And this classic example of this bacterium, Caulobacter, has all three. It has a homolog of tubulin, one of actin, and one of intermediate filaments, which are the three major cytoskeletal components in eukaryotes. So again, I'm not going to expect you to remember this, but it can use one that's really important for cell division. It can use the other to help it determine both polarity and shape. And it uses the third also to help with shape. So again, this is the cytoskeleton is kind of the bonus feature, but some bacteria can use their cytoskeletal components to help with cell division, cell shape, and cell rigidity. And these are other cytoskeletal components that help with division or even moving around plasmids. So just like eukaryotes can use cytoskeletal components to help the cell maintain shape, to help move chromosomes around, especially during mitosis, bacteria can use cytoskeletal proteins to help with division, cell shape, and moving plasmids is also helpful for division. So they have a lot of reasons that they might use this similar type of structure to what we see in eukaryotes. But not every bacteria does it. Not every bacteria has all of these. So we're almost done with the inside of bacteria. Not quite, but we're getting there. They have DNA. Remember, this is another one of those differences between prokaryotes and eukaryotes. For the most part, prokaryotes have one single circular chromosome, whereas eukaryotes tend to have paired multiple linear chromosomes. There are some bacterial exceptions, but for the most part, you're looking at a single circular chromosome that has the genes necessary for the cell to live. It is pretty tightly wound on itself. Bacteria don't use histones, right, like eukaryotes do. but I think this picture is really cool. This is E. coli that's been exploded and the chromosome has been pulled out, so that's how big the actual chromosome is. So that has to be all compacted right inside of the cell. Some bacteria may also have plasmids. That's right here. These are, again, usually circular. They can be linear. A cell can have one or more. It could be thousands of copies of the same plasmid. It could have many different plasmids. The bacteria I studied for my PhD has between 28 and 32 different plasmids, depending on which strain it is. So it's really annoying, but it is what it is. Usually, they carry non-crucial genes. Why do you think that might be? Why would you only put non-crucial genes on a piece of DNA outside of your chromosome? How many times do you forget something at your house and you get somewhere and you're like, oh, son of a... I'll walk out to my car and be like, I don't have my keys. That's helpful. Because you forget things. You leave them behind. Bacteria don't forget, right? They're bacteria, they don't think. But when they replicate, there's always a chance that the plasmid will not get replicated and transferred to the new cells. Well, I put a really important gene there. Good thing I left that behind. You're gonna die. You don't put important, you don't put your bank password on a Post-it note stuck to your computer. If you do, please stop doing that. Put your bank password somewhere else. Because I have Post-its all over my computer and my desk and the floor because they fall everywhere. You don't put important stuff that you could lose. You keep cool stuff on there though. So bacteria that have plasmids typically use them for fun bonus features, such as making a toxin or being antibiotic resistant. They don't have to do that to survive, but it can enhance under certain situations. So the thing you need to absolutely survive, you keep on you at all times. If you don't need it to absolutely survive under all conditions, you don't. So plasmids are bonus features. Then we have ribosomes. Where do we find most ribosomes in eukaryotes? Yep, the rough ER. There are some in the cytoplasm, but the rough ER is a really dense site of ribosomes. Well, we don't have ER because we don't have membrane-bound organelles. So bacteria and archaea keep their ribosomes just kind of free-floating in the cytoplasm. When we get into microbial genetics, we'll talk about why that allows them to adapt to things faster than we can. It's very, very beneficial for them. Okay. But in general, the ribosome in prokaryotes is what we call a 70S ribosome. Ribosomes in eukaryotes too are composed of two subunits, a small subunit and a large subunit. So in prokaryotic systems, we have the 30S subunit as the small and the 50S subunit as the large. So we combine those and we get the 70S subunit. And no, I'm not bad at math. I do understand that 30 plus 50 is 80. But in bacterial ribosomes, 30 plus 50 equals 70. It doesn't have anything to do with actual size. It's called the sedimentation value. And so when the ribosome is together, it doesn't sediment at 80, it sediments at 70. And in eukaryotes, it's 60, 40, 80, not 60, 40 equals 100. So, again, I don't do the naming system. Now, the ribosome, right, absolutely important. It is the site of protein synthesis. What happens when you can't make proteins? You die. Yes, that is correct. So ribosomes are a really good target for antibiotics because theirs is 70, ours is 80. There are differences. So there are a lot of antibiotics, things like aminoglycosides, tetracycline, that interfere with different parts of the ribosome and interfere then with protein synthesis. But we have to be careful because we also have ribosomes. And yeah, the 70S and the 80S ribosome are different. They're not so different that you can just flood a person with protein inhibiting antibiotics and think there won't be a consequence. One of the other challenges is that, like I told you, we're going to talk a little bit more about that mitochondria used to be bacteria. Mitochondria have 70S ribosomes. So antibiotics, in theory, can also stop the mitochondria from functioning. What happens if your mitochondria doesn't work? You die. That's where you make your energy. So we have to be a little bit careful with protein synthesis inhibitors, but they do take advantage of some of those differences. And then we have some bonus features. Some can store various forms of carbohydrates for a rainy day. We do the same thing. We can store energy for a rainy day, but we tend to store it as fats rather than as like carbohydrate inclusions. Some bacteria will also store phosphate, especially if they live in a phosphate limited environment, like marine bacteria are really classic for storing phosphate because they need phosphate to make their phospholipid bilayers and to make all of their nucleic acids because of the sugar phosphate backbone. So if you're in a resource limited environment, you try to store that resource whenever you come across it. So they'll store inorganic phosphate. Some bacteria do carbon fixation, so they can sometimes do that in a special inclusion. Some of them can detect the Earth's magnetic field, so they have magnetosomes, which have iron oxide compounds to help them detect the Earth's magnetic field. That can also help them detoxify hydrogen peroxide. And those that are in marine environments sometimes will also have a little gas vacuole for buoyancy so that they can float. So again, not every cell has them. If a cell has one, it doesn't mean it has all of them. Many cells don't have any of these at all, but they are different features that microbes have evolved to allow them to explore and exploit different niches in the ecosystem. So the last structure we're going to talk about is the endospore. Have you guys done the endospore staining? Messy, right? Like some of you made a big mess. Okay, cool. I like it. I had one who got it on her shoe and it just like went through the whole lab. I was like, oh no. I think Dr. Kong was teaching the other lab and he was like, what happened? I was like, wasn't my lab. He knew it was my lab. It was my lab. So the endospore is this really cool structure that only some bacteria form. They are always going to be a gram-positive bacterium. Gram-negatives don't do this, only gram-positives. And the endospore is this structure that allows these species to survive for hugely extended periods of time. Like they have found viable spores in things that have been dated to be like over hundreds of millions of years old. So I'm not talking like, a couple years, I'm talking like millions of years. These things can remain viable. So they're formed under harsh conditions. I mean, harsh conditions to a bacterium may be a little bit different to us. Like Bacillus does it when they get it's a little overcrowded and I'm a little bit hungry, and they'll start to form spores. Really common in Bacillus, which causes anthrax, and the Clostridium species, so C. diff, tetanus, botulism. Those are some of the pathogens that can form spores. They're resistant to drying. They're resistant to heat. They're resistant to chemicals. They're resistant to radiation. They're like the bacterial equivalent of a cockroach. They will outlast us, right? The spores will be here. They were here before us. They'll be here after us with the cockroaches. We can get rid of them. The best way that we do that here is using an autoclave, which is high heat, moist heat under pressure. You can get rid of them using various gases like ethylene oxide. It's not easy, and we don't know of just kind of natural ways to do it. They're all, human invented weird types of things that we have to do to get rid of spores. But when you're talking about things like anthrax and botulism, it's good that we can get rid of spores because those are not diseases that any of us ever want to experience. So, again, Bacillus and Clostridium are the two genera that we talk about because they cause not uncommon human infections, right? So the process by which a cell forms an endospore is called sporulation. And one of the important things to differentiate, again, especially for those of you who might be going into clinical medicine, or even if you're interested in like ecology, the type of spore, the endospore that a bacterium forms, is a very different structure from what we call a spore in things like fungi. In fungi, spores are reproductive structures. They use them to disperse their gametes. Endospores in bacteria are used to survive harsh conditions. They're not used for replication, they're used solely for survival. When a spore, or when an endospore encounters a more favorable environment, it will return to its vegetative state, and we call that germination. And again, with some of these organisms like Clostridium tetani, Clostridium botulinum, Bacillus anthracis, that more favorable environment is our bodies. So that's not great. But it makes it very difficult to get rid of these organisms. Okay, questions about prokaryotic anatomy. This is the cursed eukaryote. There's no eukaryote that looks like this. They tried to, as much as possible, put literally everything you could find in any eukaryote in one image, which is why it is the cursed eukaryote. Eukaryote has a nucleus, so the name literally means true nucleus. Prokaryote, pre-nucleus. Okay, eukaryote. So, I understand why they do the image. I hate the image, but it's okay. So the top half is supposed to be a plant cell, the bottom half of an animal cell, or a unicellular eukaryote. Anyway, plant cells, right, have a cell wall, so that's what they're trying to illustrate here. Animal cells don't, but fungi can. So I don't like the image. Chloroplasts only in photosynthetic organisms, of which plants are included. Animal cells have some structures that are kind of unique to them. Only animal cells, for example, might have a flagellum. But I think, probably what's a little bit more important are some of these conserved cells, or conserved structures that we see in both. Of course, like the nucleus, the nucleolus, the rough and smooth ER, various cytoskeletal components, mitochondrion, that one's pretty important. A plasma membrane, ribosomes, cytoplasm, Golgi. For the most part, on this side are features that all eukaryotes would have. There are a few that don't have mitochondria, but for the most part, these are the features that all eukaryotes will have. And so we can start kind of on the outside here. Again, some eukaryotic cells, not plants, but other types of eukaryotic cells may have a flagellum. Sometimes they have multiple. The most that I can think of off the top of my head is I know a eukaryote that has 4, but they tend to all be on one side, polar. Some eukaryotes also have cilia, or could have cilia. The protozoans that move using their cilia, the ciliates, they're totally covered in cilia. But we have cells that line our respiratory tract that are ciliated epithelial cells, and they beat all in one direction up to help bring up mucus and everything it might have trapped, and then dump it down our esophagus. So they help protect us. So some can use it for motility. We can use it for that type of movement and protection. Some have a cell wall. For eukaryotes that do have a cell wall, it will always be some sort of carbohydrate, but depending on the type of eukaryote, they may have different types of carbohydrates. So plants use cellulose, fungi can use chitin, especially like the multicellular fungi, and then glucan and mannan are in the unicellular fungi, but they're all carbohydrates. Some animal cells will also have a glycocalyx. It's not exactly the same as like the slime layer that we see in bacteria, but it is carbohydrates that are secreted from the cell that are attached to proteins and lipids in the plasma membrane. So we can have those. And then of course eukaryotes have organelles. These are the membrane-bound organelles, the nucleus, the ER, the Golgi, lysosomes, vacuoles, these are in plants, mitochondria, chloroplast in plants, peroxisome. So again, these are all structures that bacteria don't have because they don't have membrane-bound organelles. They do some of these similar processes, but they have to do them in a different way. Non-membrane bound organelles, well, the ribosome, it has the same function. It's the site of protein synthesis. Again, theirs is a little bit of a different size than ours. So we have an 80S ribosome. The small subunit is 40S. The large subunit is 60S, and combined that equals 80. We often talk about the ribosome in the context of it being attached to the ER so that the protein can be synthesized and transported through the ER and the Golgi. But we require a lot of cytoplasmic proteins, and so we do have ribosomes that free float in the cytoplasm too. Just not as many as like a bacteria, because all of theirs are free floating in the cytoplasm. Eukaryotes also do have 70S ribosomes, just like bacteria. We find those in chloroplasts and mitochondria. Another non-membrane bound organelle is the centrosome and the centrioles, which are important for mitosis. We don't do mitosis, so we do mitosis, they don't do mitosis. As bacteria, no mitosis, no need. So, yeah. I'm not as passionate about eukaryotes, if you can't tell. They're all right. They're okay. But plus, I think you talk about eukaryotes in a lot of classes. So this is the one place where we talk mostly about not eukaryotes. Okay, but we're going to talk about eukaryotes one more time. Okay, so we're going to talk about, we'll talk a few more times, but today we're going to talk about them one more time. We're going to talk about eukaryotes today in the context of where did they come from? And the reason I'm okay with this conversation is because they came from prokaryotes. That's why I like this conversation. So we talked a little bit about the history of microbiology, Big Bang, planet, no life, life. We estimate about 3.5 to 4 billion years ago, the first simple cells arose. And of course they were anaerobic because there wasn't free oxygen in the atmosphere. Approximately 2.7 billion years ago, oxygenic photosynthesis evolved. So photosynthesis that produces oxygen as a byproduct. Some bacteria can do photosynthesis and they do not produce oxygen. So oxygenic photosynthesis evolved about 2.7 billion years ago. A lot of the evidence for this date actually comes from geologists, because apparently you can tell when oxygen entered the atmosphere because of how it interacted with elements like iron that were on the surface. Then relatively quickly, in the grand scheme of things, we believe the first eukaryotes evolved. So I'm going to tell you kind of the order and explain why. So the order of organelles appearing, we think first was the nucleus. So scientists believe that the nucleus came first. Why? Because every eukaryote has a nucleus. So that's the defining feature. You are not a eukaryote if you don't have a nucleus. So the nucleus probably came first. If everybody has something, it had to come early. And it's thought that, the cell started kind of folding in the plasma membrane and eventually surrounded their genetic material. And we do see some prokaryotes that have this kind of intermediate type of structure where they have put a membrane with their nucleotides, but not the same as a nucleus. So we can kind of see this like protonucleus in some of our prokaryotes, which again suggests that the nucleus came first. So then you have a nucleus. But where do you get other organelles? So early cell, the idea is, if you're like picturing a little, well, I can't draw, but I'll try. Ugh, okay, black, here we go. So you have your early cell, and he's our last universal common ancestor. So we call him Luca. So, Luca, right? I'm evolving. This is great. This is fun. And then we kind of split and go this way. So these, bacteria. And then these split again. Archaea. And then somewhere here came the nucleus. So sometimes people get fussy about saying prokaryotes as a taxonomic classification, because if you say prokaryotes, right, it creates what's called a paraphyletic group. You exclude one of the descendants of the common ancestor. You can't do that. That's why reptiles isn't a real thing either. Because you exclude, if you say reptiles, you're excluding birds, but they all have a common ancestor. So you can't say reptiles. I'm just kidding, you can. Okay, so nucleus. Then, the prevailing hypothesis or theory, the prevailing theory, because this is a theory, is that one of these primitive eukaryotes engulfed and formed a symbiotic relationship with a bacterium that was capable of doing aerobic respiration. And that is our mitochondrion. And that was great, because if you can do aerobic respiration, you generate as much ATP as possible. So now you're really efficient. And then some of these got a wild hair. So we can start splitting, right? Here's our, start splitting out here. Animals, right? And over here, we'll go like that. One of these bad boys was like, yes. And it engulfed and formed a symbiotic relationship with a photosynthetic bacterium, and that became the chloroplast. And then you have eukaryotes. So broadly, that's the endosymbiotic theory. Bacteria and archaea split. The lineage that was left developed a nucleus, eventually engulfed an aerobic bacterium and formed a symbiotic relationship with that aerobic bacterium that became the mitochondria. And some of those that got a mitochondria, at least one, took in a chloroplast. Oh, photosynthesis. This opens up a whole new way to live. And that became all of the photosynthetic eukaryotes. Okay. There are other examples of basically this kind of happening in real time, but we are seeing these other types of symbiotic relationships where other organisms, for example, there's this protozoan that lives in termites. It digests cellulose to provide it nutrients. The protozoan can't live without the termite, and the termite can't live without the protozoan. So at what point is this termite now, or this protozoan, basically an endosymbiont? When does it become basically like a part of its gut rather than a organism in the gut? We're seeing this kind of happen in real time. Okay, and they're cilia. Okay. We're seeing ectosymbionts. There are some protozoans that host spirochetes, and it allows them to move. So they use them as like extra little motility structures. So we're seeing it kind of happen in real life, like in our time. But let's go back and talk about where did we get eukaryotes, and this is where we'll finish today. So this is the theory, and remember in science, like I know like in the public people are like, oh, it's just a theory. There's no like just a theory in science. A theory is, there's very few laws. Thermodynamics, gravity, right? Everything else are theories, which mean they have a ton of support. And no experiments that anybody has ever done have been able to disprove them. These are theories. So the prevailing theory is the endosymbiotic theory, that nucleus, then mitochondria, then chloroplasts. So what are some lines of evidence that support this? Well, mitochondria and chloroplasts are very similar size and shape to their theorized bacterial ancestors. Okay. Bacteria, mitochondria, and chloroplasts all have a single circular chromosome. What do eukaryotes have? Multiple linear chromosomes. So they have very different genetic material. Bacteria, mitochondria, chloroplasts all have a 70S ribosome. What does the rest of the eukaryotic cell have? 80S. So different ribosomes. The bacteria that we think became the mitochondria and chloroplasts have a double membrane. So do the mitochondria and chloroplasts. They have a double membrane. And the proteins in that outer membrane of the mitochondria and chloroplasts are homologous to bacterial proteins, not eukaryotic proteins. They reproduce autonomously by binary fission. And by they, I mean bacteria, mitochondria, and chloroplasts. They don't do mitosis like the rest of the cell. And those spirochetes that are the ectosymbionts on some of those cells, they can move independently, meaning it's not an organelle under the control right? The cell does not control the symbiont, but it is dependent on them. So there are lots of lines of evidence that support prokaryotes leading to eukaryotes. So this is a really important theory for you to keep in mind and think about. And I will see you all on Tuesday, where we will start chapter 5.